This week, our program looks at cheese making, the preservation of foods, and what you should know about a molecule essential to life, cholesterol. Did you know that the great variety of cheeses is due in part to the great variety of milks? Like wines, milk too has its vintage years. But what makes the difference between a nice firm Gruyere and a soft Camembert is mainly the process of fabrication. Over 1,000 years ago, man discovered that the best way to preserve milk was to convert it into cheese. Because it is drier and more acidic than milk, cheese is more resistant to spoilage. In addition, it concentrates the main nutritional elements of milk: lactose, which is a sugar, proteins, the main one being casein, and lastly, fat. There was a great variety of cheeses. Their flavor, aroma, and consistency vary depending on the way they are made. Fresh cheeses, for example, are unripened and moist. Others, like Brie and Camembert, ripen from the outside by surface molds. Blue cheeses, such as Roquefort, are ripened from within by veins of mold, which give it a distinct flavor. Other cheeses like Beaufort, Emmental, Gruyere, and Comté are heated, then pressed, which prolongs their shelf life. Let's take Beaufort, for example. According to the traditional fabrication method, the cows are milked out in the open, in the pastures. The milk is taken straight to the cheese dairy, where it is poured into huge copper vats. It is heated to 32 degrees Celsius, the ideal temperature for the lactic bacteria in the milk to multiply. This temperature also ensures the milk will curdle. Curdling results from stirring a coagulating agent called rennet into the milk. Rennet is extracted from the stomach of a milk-fed calf in brine. Rennet is an enzyme that coagulates the casein. Casein is the main protein in milk. It is composed of micelles, which are spherical particles. The micelles all carry a negative electrical charge, so they mutually reject each other and remain separate. The rennet, however, causes the micelles to clump together. This phenomenon results in the formation of a gel, the curd. When the curd is firm enough, the cheesemaker cuts it into small chunks no bigger than a grain of wheat. This step, the cutting of the coagulum, allows the whey to drain out. Whey, commonly called buttermilk, is a liquid made up of water, residual proteins, fats, and mineral salts. The curds are then cooked and agitated, which makes them denser and more compact. Once the cooking is completed, the mixture is sucked up into a drawing-off bell. The curds are then gathered in a cloth, as is the tradition. It is then pressed in molds for 24 hours, during which the grains knit together and all the moisture is eliminated. The cheese then takes on its definitive form. During the pressing, the bacteria transform the lactose, the milk's main sugar, into lactic acid. This acidification is a crucial factor in preserving Beaufort cheese. Next, the cheese is taken from the mold and immersed in brine. The salt hardens its surface, promotes the formation of the crust, and prevents the development of undesirable microorganisms. After 24 hours in the brine, the cheese is put into cool, damp caves to ripen. The ripening process consists of salting each wheel of cheese 2 or 3 times a week for over 6 months. The salt helps preserve the Beaufort and gives it its characteristic taste. Each wheel is then rubbed with fabric to promote the development of surface microorganisms that will contribute to the ripening process. During its long stay in the cave, the cheese undergoes various transformations. For example, the casein molecules split up, producing amino acids, which give the cheese its aroma. Some cheeses, such as Emmental, are ripened in warm caves where the bacteria in the milk will produce carbon dioxide. This gas, trapped inside the cheese, is what forms the characteristic holes. On the other hand, in cheeses without holes, like the Beaufort, the cooler cave temperatures keep the production of gas in check. Today, much research is being devoted to improving the quality of cheeses. For example, researchers are currently putting together a collection of bacterial strains presenting varied genetic potential. They gather natural ferments from various cheese factories. In a lab, they study the qualities of the different strains. At the end of their testing, only the best strains are selected and kept in reserve. They are then made available to the cheesemakers, who can choose the strains best suited to the type of cheese they produce. Scientists' efforts combine to maintain a tradition the gourmets of the world would be loath to see disappear. There are several ways of preserving foods: salting, freezing, dehydration, and of course the good old tin can. Unfortunately, most of these methods take away from the freshness of foods. New processes may remedy that problem. Today's consumers no longer have the time to do their food shopping on a daily basis. But they still want fresh, healthy, natural, additive-free, and preservative-free foods. Though tried and tested, conventional preservation methods have their drawbacks. For one, they alter the freshness of foods. New processes now prolong the life of food products while keeping them fresh at the same time. One such process is controlled atmosphere packaging. It consists of introducing a given gaseous mixture into a food's packaging. The gases reduce the rate of microbiological growth and chemical action that normally cause a food product to spoil. The first step consists of de-aerating the packaging to rid it of all oxygen. Oxygen is the main cause of food spoilage. Oxygen, for example, will turn fats rancid. It will also cause the proliferation of certain microorganisms. Oxygen is also responsible for making certain vegetables and fruits turn brown when cut. The change in color is due to the action of an enzyme inside the cells of certain plants. When these cells are cut in the presence of oxygen, the enzyme causes browning. Vacuum packing eliminates these many inconveniences. The second step consists of injecting a gaseous mixture into the packaging. A mixture of nitrogen and carbon dioxide is generally used. Nitrogen is an inert gas, which means it does not react with the food product. Its main role is to inflate the packaging to protect the food from being crushed. The carbon dioxide reduces the rate of mold and bacterial growth. It dissolves in the food's water and becomes carbonic acid. Carbonic acid slows down the development of microorganisms. Though generally an undesirable element, oxygen is sometimes included in the gaseous mixture. For instance, it is used to maintain the bright red color of beef. The meat's color comes from myoglobin, a pigment found in muscle fibers. This pigment's role is to store the oxygen that muscles need in order to contract. When deprived of oxygen, myoglobin gives meat a brownish color. Adding oxygen to the packaging helps beef to retain the rich red color consumers are so fond of. As for raw vegetables, even peeled and cut up, they remain living organisms and continue to exchange respiratory gases with their environment. In order to preserve their physiological activity, they are packaged in a permeable film that allows them to breathe. The film lets oxygen into the packaging. At the same time, it allows the carbon dioxide produced by the plants to escape. All methods of food preservation will alter the quality of foods. Consumers are particularly sensitive to variations in texture and crispness. To measure these qualities objectively, researchers use a penetrometer. This device gauges the penetrability of a food, simulating the effect of a tooth. Restaurants also benefit from the advances made in food preservation. Thanks to cellopack cooking, restaurants can offer their customers a multitude of elaborate dishes at all times. But more importantly, they can serve them in record time. The food is pre-prepared in the laboratory before it reaches the restaurant. Each food is processed in a special way. For example, raw fish and meat are cut into individual portions and seared briefly on a grill to give them the familiar grill markings. They are then placed into plastic bags that are vacuum sealed. All vacuum-packed food is steamed or cooked in water at a temperature above 65 degrees Celsius. This type of cooking eliminates most of the bacteria. It also preserves the flavor of the foods and prevents drip, the loss of their juices. Once cooked, the vacuum-packed foods are put into quick cooling chambers for 20 minutes and brought down to a temperature of between 0 to 3 degrees Celsius. They can now be kept for 14 to 21 days. They are delivered to the restaurants in refrigerated trucks. The restaurant only has to heat up the various foods making up the diner's meal, remove them from the bag, and put them on his plate. The customer will never suspect that his gourmet meal was prepared 2 weeks in advance. A few years ago, high cholesterol levels were discovered to be linked to a greater risk of heart disease. Almost overnight, a myriad of cholesterol-free products appeared on store shelves. Today, many scientists are not convinced that a cholesterol-free diet will affect our health one way or the other. In most industrialized countries, cardiovascular disease is one of the main causes of death. The high rate of heart disease is often ascribed to nutrition. Today, cholesterol has been singled out as the main culprit. And yet, cholesterol is a life-sustaining substance. It plays an essential role in building cell membranes. It also plays a role in the body's synthesis of innumerable hormones and vitamin D. The cholesterol present in the body is mainly found in the bloodstream. It comes in part from what we eat. The foods highest in cholesterol are egg yolks, organ meats, shellfish, and butter. Conversely, vegetables and fruits contain no cholesterol. However, most of the cholesterol in the bloodstream is produced by body cells, liver cells in particular. On its own, cholesterol cannot circulate in the bloodstream because it is insoluble in water. It becomes soluble by combining with large molecules called lipoproteins. Some of these particles are known as very low-density lipoproteins. Their role is to ferry the cholesterol to the cells, where it is either used for energy or stored for future use. However, when there is an excess of very low-density lipoproteins, they cling to the walls of blood vessels, where they deposit their cholesterol. If an artery presents tiny lesions caused by high blood pressure, for instance, the cholesterol may infiltrate into its interior walls. In time, small blood clots and layers of sediment may join the cholesterol, forming an atheromatous plaque. As the plaque grows bigger, it gradually narrows the artery and blocks the blood flow. If the atheromatous plaque forms in one of the coronary arteries, the heart muscle will be less irrigated. The ultimate result is a myocardial infarction, or in layman's terms, a heart attack. However, there is another type of lipoprotein, the high-density lipoproteins, which play a cleansing role. They pluck the cholesterol from the artery walls and carry it back to the liver where it can be excreted. These cleansing lipoproteins are nicknamed good cholesterol. Very low-density lipoproteins are referred to as bad cholesterol, since they contribute to atherosclerosis, the development of atheroma plaques. A number of strategies have been recommended to lower blood cholesterol levels. One suggests reducing total dietary fat intake. In response to that recommendation, grocery stores are now inundated with light products. Preparation of these products may simply consist of eliminating the fat, as is done with cold cuts and ham. For dairy products, such as cheese and yogurt, the fat is first completely removed from the milk, then added back in the desired amount. A second strategy to lower blood cholesterol levels consists of reducing the intake of foods high in cholesterol. Low cholesterol food products have also made their appearance on the shelves of grocery stores. But according to the scientists, any diet aimed at lowering cholesterol levels should recommend a lower intake of saturated fats in favor of polyunsaturated fats. Saturated fats raise the level of cholesterol in the blood and facilitate its deposit on the inner walls of arteries. Saturated fats are found in meat and dairy foods, and in some plant products, notably coconut and palm oils. Polyunsaturated fats, on the other hand, lower cholesterol levels. They are found in most vegetable oils, such as sunflower and corn oil, as well as in fish. There is, however, increasing doubt about the effectiveness of diet in solving cholesterol problems. We know that blood cholesterol levels depend more on the liver's production of cholesterol than on food intake. In fact, only 20% of the cholesterol in the bloodstream comes from food. Moreover, high cholesterol levels are evidently linked to genetic factors. Researchers extracted the DNA from the blood cells of several people. Among the samples, they retraced various genetic types conducive to the formation of defective lipoproteins. These lipoproteins have difficulty binding to the cells that require cholesterol. Rather, they tend to build up in the bloodstream and are therefore more likely to leave their cholesterol on the walls of blood vessels. While it has been proven that high cholesterol levels increase the chances of cardiovascular disease, it is still not clearly known what impact diet may have on the prevention of these diseases. Dietetics is a science, but like many others, it does not always manage to thwart passing fads or ill-founded fears.